So I apologize for the 2-minute delay. But I was teaching from 10 to 12, so this is my third hour. Bon. Ahla. So today we're gonna, so basically you're all experts in visual system, somatosensation, and a little bit of basics, and right, or more even, I don't remember. Now, let's see. And now we're gonna end the course with the two last sensory systems that we're gonna talk about. First one is the olfactory system. I'm gonna give two basic lectures. We're going to have one advanced lecture from Yoram Ben Shaul about the Vimero nasal system, and then some papers, and then we're going to end with the auditory system, which I will also be giving, and we're going to have one advanced lecture by Eli, and finalize the course with two series of lectures. So one is about the auditory, and the other one will be combination of auditory and plasticity, which has to do with plasticity in sensory systems. Yeah, and I still owe you guys the auditory lectures. The papers of the auditory, and of course the last one, but we still have time there. I have to see what I'm going to teach. So it will depend on that. Okay. And I'm still working on what I want to put on the lecture. So it depends on what I will manage to fit in today, and then I'll put the lecture on the web. Or I can put it now in the Hafsaka, and then I'll update it as you wish. Okay, anyway. The sense of smell, also known as olfaction. It is one of the most ancient senses, runs long and deep into biology, into most animals of biology. Humans are not particularly great smellers. I mean, if your nose is clogged or you cannot smell, if you're anosmic, then, life quality is not deteriorated, certainly not as bad as vision or audition, which really impairs the quality of life. However, if you're a mouse or a dog or a worm or a fly without the olfactory system, you're in serious trouble. In fact, they cannot really live well without this sense. It's a very central sense, and in some ways, because in neuroscience, our experimental animals are using this sense to a large extent behaviorally, and because we really want to understand the interactions between the brain and the neural circuits and behavior, this is being realized as an increasingly important sense, although still it's not as, you know, in the biomedical research in terms of funding and money and things like that, it's quite difficult, more difficult to get money in the context of olfaction as opposed to vision and audition, which is more easily, one can easily sell it as a very central biomedical issue in human beings. Okay, but for really understanding very basic concepts in neuroscience between neural circuits and behavior, this is as good as any other sense, if not more, for sure, for animals like a worm, which is a very central sense. Also, for human beings, it's unique in several senses. First, it's in several aspects. First, it's used. There's a lot of money around olfaction, especially in the perfume industry, especially in the food industry, where lots of money is spent on making the odors smell good and making, you know, the coffee smell good. You do know, I hope, if not, then you should know that the instant coffee that you drink at home, it's a lie. It's a complete lie. They take the coffee beans and they grind it, and they make it dry, and it's completely odorless. Okay, if you go to the factory, the coffee, the taster's choice, and the Folgers coffee, and all those, it's a complete lie, because when the coffee is ready, it's odorless, and then they add upon that an artificial odorant, which smells like coffee. Of course, it also smells like morning and love and etc., but basically, it's a lie. The real coffee is the Turkish grind coffee, where you take the beans, you grind it, and smell it, but the instant coffee is a lie. There are some people who are experts in smelling, you know, all those wine tasters, etc., also known as the noses. They get a lot of money for their work. And again, a lot of money is rolled around the industry of food and perfume. However, there's very little connection between the perfume industry and the real neuroscience. They don't really want to understand how is it happening and how do we perceive. If you go to a chemical senses meeting, very two separate groups of people, ones who are regular neuroscientists who want to make the connection between neural circuits and behavior, while the other wants to sell and get more money, etc. And sometimes the gap is so big, which is a little bit funny. Okay, we're going to talk mainly about rats, rodents, but not only, because there we can dig into the nervous system and really understand better what is it all about. The sense of smell, like I said, runs through the whole animal kingdom. This is a classical example of a hound, the classical example of a hound dog, which is going after a dead rooster here, which was dragged by the yellow line through the field. And you can see so basically the rooster was dragged from here and then taken to a left side, and the dog was, or just through the field here, and the dog, we can look at its pattern of sniffing to find the rooster. If you've ever went recently to the airport, you can see all these great dogs that are sniffing around, and their capacity to find odorants in a high background and a high noisy environment is really remarkable in the animal kingdom. We have just 2 years ago, we went here to the police department. They have a dog academy here. Actually, it's in the north. Here they bring the final graduates of the academy of the dogs to find bombs and hashish and drugs. And we got a demo of the dogs. It's unbelievable. They take a gram of hashish and they bury it in a tree, which is 1.5 meters above the ground. They let the dogs, from a field of 200, 300 meters look for it, and they find it in 100%. So the accuracy of some animals and their ability to use smell, and everyone who has a dog, for example, knows this, that they are continuously smelling, and this is a very central sense. Of course, they can smell very tiny amounts of chemicals, and how they do it and how the brain decodes this information is what we're interested in the context of this course. This is a very nice picture that was taken by Noam Sobel when he was now in Weizmann, but initially in Berkeley. This is one of his graduate students where he wanted to make the point that they can smell. In this case, it's a lawn in the Berkeley campus, and he took a chocolate, Nutella, and rubbed it on the grass. And the student is working for money, of course. If she can follow the chocolate on the grass, and you can see he put gloves so she can't hear and can't smell and only smell. All the other senses are discarded, and she can follow the.
And it wasn't amplified, the smell, like she didn't have anything amplifying.
No. She could define the smell without a problem. Not without a problem. It's much slower than this animal, of course, yes, in terms of the capacity. But when you talk to Noam and the people who work with human olfaction, so there is a dichotomy. They are arguing that indeed the human olfaction is a very important sense, and but clearly it's not comparable to the abilities of other animals in this, at least in this context. Okay, and we'll touch upon that as we go along. Okay, the first experiment I want to show you is from 1976 about imprinting in salmon fish, odor imprinting, just to show you again the impact and capacity of odors. Basically, this is a field study done from the University of Tennessee, I think, or something like that. Basically, they took 3 groups of fish, of salmon fish, which were raised in a hatchery, huge tanks of water. One was a control group. One was raised with an odor which is indicated in the study M. I don't remember exactly the name. And the other one is PA, I think phenylanel something, whatever. This is one odor. This is the other odor. And when they were young, they grew in these fish tanks with this odorant. Basically, it is an odor imprinting. And then they released them in the Lake of Michigan in a given point. And in the Lake of Michigan, there are 19 small creeks that are around the region where they did the experiments. And they released the fish here a year and a half later. And as you know, salmon are homing. They go back to the creek where they were born to spawn and then die. And basically, they released the fish here and had students all over the 19 small creeks. And 2 creeks were manipulated. One creek was manipulated with the smell of M. Here, I think this was this creek up here, and the other creek was scented with the smell PA. And then they counted how many fish they could recover, again, a year and a half later, after they were imprinted initially, and you can see the results here. I normally don't like to show tables so much in lectures, but again, this really shows you the dramatic types of results. This is a science paper or a nature paper. You can see that, in Stony Creek, which is over here, they found 1 fish, and they could tag them. In the control, there was no tag. In the fish that grew up with the odor M, they chopped one of the snapirim, the fins, in a given way, and in the other group in another way. And you can see that they could recover a few fish as noise in the other creeks, but 207 fish were recovered where they actually put the M odor in this specific creek. And the PA, they found most of the fish in the other creek where they released the PA, and in the control group it was all over the place with some bias to these two creeks, but nevertheless you can see the impact of these types of results. Okay, so odor imprinting in fish, it goes from fish to humans, and this is a paper I read actually this morning, which was published a few weeks ago in Science by Andreas Keller from Colombia, I think. And there is a notion, if you read in the newspaper and in the Candel book and other places, they will tell you that humans can differentiate 10,000 odorants. That's like the common general public knowledge of odorants, and they claim nobody actually really tested it in a more rigorous way. So what they did here is they did an experiment where they took mixtures of odors. They took three types of experiments, one with 10 mixture of 10 components, out of 128 possible odorants in their pool, which are shown here as the green dots, mixture of 20 components and mixtures of 30 components. And they did these mixes. So basically, they took from, let's say, from the pool of 10 mixtures, they took, and they mixed and matched and tested whether a person, a human being, can discriminate between the two different mixtures, mixture A and mixture B. And the more overlap there is, of course, you can take 10 odorants in a mixture in two different, with no overlap whatsoever, and that would be a very easy test for a human being to differentiate between two different mixtures, and then you can increase the overlap between the two mixes. So for example, the black bars are where there is overlap. In this case, 3 of the odorants overlap and 7 don't, so there's a 30% overlap, 60% overlap, 90% overlap. They did this with 20 mixtures, 20 components in a mixture and 30 components in a mixture, and they can get different values from no overlap at all, 25, 30, 33, 50, 60, 66, 75, 90, 95, and 97% overlap between the mixtures. And as you can imagine, the more overlap there is, the harder it is to differentiate between these between the two mixtures. Basically, the data is shown here. Actually, they had three alternative choices. So basically, the choice level is at 30%. But you can see that as So as this is chance level, as you go to 90%. Let's look at here, 96% overlap between the mixtures. No person could discriminate such a high overlap. 95%, none of the people could discriminate the overlap. 90, there were already some that could differentiate. This is in the red arrows. And of course, as you make it in each group easier and easier, you can get better scores. The bottom line, and we're not going to go into the calculations here. Again, this is just an introduction or gimmick before we go into the nervous system, is that while until a few weeks ago the previous estimates was about 10,000 odors that we can differentiate, now they claim that we can get trillion odors, 10 to the 13 or something, human beings can differentiate in terms of different mixtures. And in the literature, there are a few older papers looking at the colors and tones, and the single tones and the colors, or the color hues, was this was a few million, I think 7 million, and about 400,000 different tones. So the claim here is, odors are at least as good as tones and colors. Of course, once you start to do different intensities, different combinations, the numbers will rise as high. But again, the point here in the paper is that human beings are not limited in the capacity to smell so many odors. Okay, it's just a recent paper they plugged in the talk this morning.
It was without any training, right?
No training. You get there's some. I think they got 200, and each person got 250 trials, and different mixtures or something like that. It was actually well controlled. Looked as if it was well controlled. Yeah.
So here they compare pure tones against mixed.
Yeah, it's not a true comparison. So what?
Kind of unfair comparison.
No, it's not. They say that this was the comparison initially, that odors were about 10,000, and in a comparable study, and I'll show you why, because it's a little bit complicated. Okay, it's easier to do this in tones and colors because you have an axis that you can more easily compare. And here they say, okay, so the previous estimates were 10,000, but real odors are mixtures, and we are, in the trillions. And of course, these will also go up to the trillions if you start to do images and things like that. But the point here is that, we'll let the visual neuroscientists do these calculations. At least let's fix this graph and put the odors, many orders of magnitude higher, and we'll let the auditory people figure out how many sounds they can discriminate. Yeah, it doesn't mean that smell is better than sound, of course, or you can do it more with that. It just means that it was highly underestimated before. That's the point of this paper. Okay, another just point to a teaser. If you look in the Candel, this is not this is actually updated on the earlier version of Candel. But there are 8 pages on olfaction and 100 pages on vision. So again, you can see the imbalance between what we really know about the sensory system. And this is starting to change. So if you look in a paper from 1965 by Pitts, Letvin, and Gestland, whatever, and J. Physiology, which was a very respectable journal, talking about olfaction. Just to quote, let us suppose in the very limit that we have utter chaos. We know basically nothing about the olfaction. In 2006, from Wilson and Mainen in Annual Reviews, the study of olfaction processing may no longer be in its infancy, but it has yet matured beyond stormy adolescence. Okay, so we are really making our progress high. We call it job security in the field of olfaction. Okay, we don't know. We have enough that we don't know to study, and what I'm going to show you is that we basically know something about the first 2 stations of olfaction, maybe a little bit on the third, but not much beyond. And like in any other sensory system, I like this image because we really want to understand the conversion. Basically, in this course, this is what we do. We make the conversion between, or try to understand from the stimulus itself to the perception. This is one of still the holy grails in any sensory system in neuroscience, and here too. And olfaction has its own niches, which is unique. It goes directly to the emotional centers without going to the thalamus and higher processing, things like that, and lots of anecdotes. Okay, I have a family member who is a psychiatrist working with a lot of people in depression and under all kinds of traumas, and the smells are something that run very, very deep. People that come from wars, from all kinds of catastrophes, they cannot get rid of the smells. Okay, especially from people coming from wars. He has patients who are dipping their hands in Javel, in Economica, just to get rid of the smell, so they can smell the HCl strongly. They dip their hands in Economica, and all days they can smell their hands because they can't get rid of the smell from the memory. So there's something that runs very deep between odors and emotions on one hand. On the other hand, as you will see with Yoram Ben Shaul, I hope, when he will give you the talk about the vimoronasal system, you see all these commercials about male putting a deodorant and all the ladies are running after them. This is basically nonsense, and because human beings don't have a vimoronasal organ, and you will see that in mice it's real. Okay, there are some chemicals in flies and worms and mice, etc., that induce attraction very strongly, sexual attraction as well, and the femoral-nasal system in humans almost disappeared. But Yoram will talk about that when we will get to the pheromone system, which is both a separate and an overlapping system to the general odor system. Okay, so like other senses, the odor stimulus is normally, natural odors, natural smells are normally complex. And we know, a rose is, I don't know, 275 different chemical components. Urine is 135. I don't know. Every odorant has its own signature. And the question is, how do we know what's in the mixture? There are ways to do that. Okay, each one and the mixture are overlapping. Again, not very different than other auditory stimuli, visual stimuli, which are similarly as complex. The problem is that in vision and audition, we have a way to quantify this on a meaningful scale. And in the olfactory system, it's much harder to quantify it on a meaningful scale, basically because we don't have, I would say, the good relationship between the biologist and the chemist. It's a big gap between us. There are machines today that can detect in a very, very high quality, very small traces amounts of chemicals. But there is not a lot of connection. Like for me, it's very easy to, on one hand, so this is one hand. On the other hand, it's an objective thing that one cannot really, we don't really understand the nature of odors, that we can transmit this information very fast. I can't have a colleague on the other side of the world and ask him to, smell that. It's impossible to transfer smell. So people are working on electrical noses and things like that, but it's not really possible at the moment to do this very easily. Audition, I can in a click send my colleague in San Francisco an image or a sound file, and he can recapitulate it in his experiments in a 100% fidelity, very easily and reliably. Odors are much more complex. Also, for someone like me in my laboratory, which we use, or we are interested in how the brain processes natural sounds and natural odors, then to extract the natural odors is much, much more difficult for us. Okay, for example, we study, I don't know if I showed you the first lecture about the interaction between the babies and the mothers. So the babies, they call the mother. They have all kinds of vocalizations. One can get a $10,000 microphone with some good amplifiers, put the microphone next to the baby pup. When he's calling, mommy, mommy, you can extract this. We have banks and databases of exactly the sounds that the biology played to us. But now I want to understand the smell of the pups. That's not an easy task to extract the smell of the pups. One can do it. There are all kinds of carbon electrodes that one can put in the cage, so they will absorb the odorants in the air and then can take it to a GCMS machine. And people do that, these kinds of things. But again, it's not as easy as the study of other senses. Nevertheless, people have in many years tried to quantify odors and put them on a scale. It started, as usual, with psychologists. And initially, it was a scale of 7 different values to an odorant: champa, forest, musky, floral, peppermint, ethereal, pungent, putrid, etc. But then there were big fights between the psychologist and another group of psychologists. This guy came up, no, it's not 7 categories, it's actually 9 categories, very important. And now there are 9 categories of different, that one can perceptually describe an odor. And then later on, the new trend of the new psychologist was not a list of one of these. You cannot only categorize 1 out of 9. It's actually a prism of odors, and one has to put you know, how much spicy this odor is versus how much resinous it is and burnt, etc. And each odor is on this kind of odor space. Okay? And we didn't make huge progress from then. The only others, when you start to go into the nervous system, try to categorize the odors based on the chemistry, actual chemistry. And if you look in a chemistry book, one can describe a single chemical by over 1,500 different descriptors, which will describe that chemical, okay? A melting point, its size, its chirality, all kinds of things. So in some papers, you will see people who are trying to take one of these parameters. For example, in this case, the carbon number, for example, you know, how many carbon chains there are in a given molecule, and quantify, put it on a linear axis, that 3 carbon or 4 carbon is more than the 3 carbon, and the 5 carbon is twice as much as the 4 carbon, etc. So to try to get this on a similar axis. Yes?
Sorry that I'm making the same point again, but if you say that sound and vision stimuli can be described by a few physical continuous variables, it means that you are talking about pure tones, or one color is completely homogeneous images, which would be analogous to just taking one chemical substance, right? And if you talk about real world stimuli, then in sound and vision this would be complex sounds and very complex images, and then you...
No, so I'm talking about the discrete units of, so if you talk about pure tones or even natural sounds, you can break them into several components and reconstruct the exact pure tone with a few different components, okay, in the temporal and the spectral aspects of the sound. In odors, the basic component of the odor is a chemical, is a single chemical. And now if I want to compare whether butyraldehyde, how different it is from pentaaldehyde, okay? So this has 4 chains and this has 7 chains, and whether the one who has 10 chains, the octaaldehyde, is as twice as much as the butyraldehyde, it's not a trivial thing to do, I think. And it's not comparable to taking, you know, even a more complex sound and saying this is twice as much in the spectral axis than the previous tone, or in the intensity, or in its FM structure, or in its AM structure. And that's easy to do. And here it's not. And these are the pure tones of olfaction. Okay. Yeah. You can smell only volatile molecules, not necessarily, but mostly are organic molecules, yes, that we are using in olfaction. So I don't know completely the answer to that, you know. Okay. Anyway, so unlike other stimuli, which experimentalists, etc., can break down rather easily, olfaction has some new challenges. That said, again, Noam Sobel from the Weizmann Institute will tell us that this is wrong, and we can, in fact, he has a way to simplify the basic any odor into a single axis, and this axis is an axis which he calls compactness, how compact is a molecule. And the way he did this is basically take a survey of reports from human beings on a given axis, and he shows that there is you can put any odor on one axis of pleasantness, from the hedonic value of an odor, from a very bad smell to a very good smell, and this is one axis that all odors share. Yeah.
This would be correct across different cultures?
Across different cultures to some extent, yes. Normally in those papers, one does give different cultures. If you want to make a point, you have to show it in different cultures, and there will be differences, even within individuals, as we will see soon. But generally, yes. His claim is it's all over the world. One can put a single axis of pleasantness, and mostly most people will tell you that a rose will smell on the good side of this axis, and I don't know, ricavon will smell on the other side of the axis. Ricavon, how do you say ricavon? Broth, yeah, something like that will smell bad on the other side of the spectrum. Yeah.
What about smells that we know that there are individuals that they like fuel, gas or something?
Yeah. Okay. So again, I don't know the answer completely to this question. I will show you one example of androstenone that basically the community is split into half, and we found an explanation to that, but some think it smells bad, some think it smells good, and we know why, but I don't think anybody knows the answer to this question. But there will be, differences within a spectrum, and if it falls on the other side of this 50%, it's...
But also that we can say, about sound. Like I could like some sound, and other people.
Yes, but generally speaking, low sounds with a low spectral components are more pleasant, and if you play, ultrasonic or high at the high kilohertz sounds, they will be less pleasant normally. Okay, so again, on this axis, people can change in between based on experience, etc. But in culture, of course, which is part of experience, but generally speaking, there is an axis. In this case, Noam claims this is the main axis and no other axis matters. Okay? Okay? Yeah, let me just finish. Okay, what he did here, so he correlated he used PCA to look whether one of the chemical descriptors of those chemicals that were scored on the pleasantness axis can describe most of the variants. And what he finds is that a measure of odorant structure that is called its compactness is nicely correlated with pleasantness. And this measure is something that would tell nothing to you, and I would bet also nothing to any chemist who looks at these books, because again, there are 2,000 different descriptors of each molecule. But basically, Noam claims it's not so complex. Life is very simple, where there's a one axis of pleasantness, and it can be described by compactness, and he's trying to push this concept very, very strong. Yeah, question?
So what's the functional use of having this compactness or pleasantness or whatever kind of scale for smell?
For
smell. So what's the use of describing smell in terms of compactness, whatever this compactness may mean?
No, so I want, as a researcher, I want to know if I give smell X, if it's 2 times stronger than smell Y, okay? I want to study intensity. So in auditory system, it's easy. I turn on the volume twice as much on the intensity, which is a decibel value. In this case, it's not a linear one. We will talk about that, but it doesn't matter. And I can really perceptually know that a given sound is twice as much as another sound. And then I can test how is intensity coded, for example, in the brain. In chemicals, actually intensity is a linear scale, so that's not a good example in this case. But again, you need some kind of descriptor to understand how far is one odor from another, for example. And there are ways to do that. We will see them on the way, I hope, because I'm only on slide 11 out of 120, so maybe I'll have to run faster, so maybe not. Yeah. Shoot. None. Okay. Okay, yeah.
So can this axis be researched at animals also, apart from humans?
Yeah, so in one of his papers, he claims that he looked at mice, there's not a lot of human data, maybe some fMRI, but for sure mice data, he says they work well. Rat data work well also on the axis of pleasantness. It's if it's more attracting or less attracting, and fish data didn't fall nicely on his theory. So that's what I can tell.
Second question, is there an advantage to study odors in humans apart from the pleasantness axis?
for humans, like any other system. Yes, of course, you can give them instructions. Everything that has to do with psychophysics is still much better than for example, feelings. Okay, you can go into all kinds of studies of emotions, which you cannot really do with animals. Us, things like that, it's extremely difficult. Again, another nice paper from Noam, was 2 years ago when Yair Lapid, which is our Ministry of Finance now, the good-looking guy, opened the Channel 2 report after the commercial. It was Friday night, and the item was, why are scientists crying in the Weizmann Institute? That was the highlight, and it appears that they did a study where they collected tears from women that were crying. They showed them the movie of the champion, Sipuרו Shel Aluf. I don't know how it's the champ, a very heartbreaking story. They collected tears from the women, and then they gave the smell to male subjects, and the male were aroused when they smelled the women tears. This thing you cannot do in mice at all. This type of experiments. Yeah. Shoot.
On the first line, the principal component.
Ah.
Yeah, the first line on the right.
The first line of the right, yes.
So the principal component number 1, explains only 25% of the variance. So how it explains only one dimension? It's not really one dimension, right?
Again, no, it's not only one dimension, of course, but this explains most of the variance. Yeah. This was the most significant principal component.
And another question.
Yeah.
Do you have evidence that baby mice produce odor for their mother?
The baby mouse?
Yeah, not only sound. They produce sound for the mother.
Oh, absolutely, yes. Yes, okay.
They synthesize what they usually do to produce odor for the mother.
The smells that go out of their own body, and different glands produce a lot of different odorants. Okay?
Only in mice?
No. In human as well. I mean, I come from a Yemenite origin. At least 30% of me. My father eats chilbe. You can rest assured I can smell it when I go home. Okay, to my father. Your diet, things like that, can influence the way you smell, and also the genetic makeup of the animal itself. It's well known that dogs can do wonderful things, but cannot differentiate between the T-shirts of identical twins and things like that. So yes, the smell, just maybe this is very sad, the level that I can make progress here. So let's run fast. I'll tell you in some experiments that we did in the lab in this context, okay, to convince you that certainly mother uses the odors to detect her pups. Okay. So, until now it's a general introduction. And now we go into the nervous system. The nervous system starts here. Like in other senses, in the epithelial surface, this is in the oral cavity. There is a structure which is called the olfactory epithelium. And basically it is exposed to the air which we breathe. And we freely breathe all the time. So odors or the air is circulating continuously in our epithelium, and the molecules come in contact with the epithelium. Actually, there's a small layer of mucus, and there are actually neurons which are exposed to the outside world on one hand, and on the other hand, send their axons directly into the central nervous system. Okay, so these are one of the only actually receptor cells that have connection from the outside world into the brain, into the CNS. Most other senses, the receptor cell is exposed to the world, but then it connects onto a neuron that sends on the information deeper. Here there is a direct connection. If you look at this, when you take out, I assume you did not, but when you take out an epithelium from a live animal into the dish, it looks like a jelly. It's shaking. And that's because these processes, which are called cilia of the neurons of the receptors, are continuously beating, so it looks like it's shaking. It rotates like that, which is quite impressive. And basically these cells, these neurons, olfactory receptor neurons, the receptor cells, which are neurons by any means, are surrounded by supporting cells, which basically are producing all kinds of molecules to make up the mucus and all kinds of other goodies so the neurons can live happily ever after. They don't live happily ever after. They live around 1 month or so because they are connected to the outside world and a lot of toughness they have to withstand. There's actually a lot of regeneration going on, so one can find also in the epithelium these basal cells, which will be new neurons throughout life. There's some arguments in the field. Estimated lifespan of olfactory receptor neuron is about between 1 month and 3 months, and if you kill all the olfactory receptors, they will regrow if the basal cells are here. There are all kinds of proteins that shuttle the odorants, volatile odorants, to these cilia, and basically the olfactory receptors, which we will talk about soon, sit on the cilia here. Yeah.
Is all this true for humans, mice, rodents?
Not all the animals. I mean, the worms have a different olfactory, flies have a different sensory systems. In mammals, yes, they have an olfactory epithelium.
And the life of a neuron is about 1 month in all.
No, we don't know that. We know only there are very few studies in mammals, and that's roughly the number.
So can we study the receptors themselves? And so basically, if we know all the different receptors, then we can know exactly how many odorants we can detect. And then
We'll talk about that.
This is the first question, and the second question is, do they change? Like if I give different stimuli, could you produce new sensors or new?
Okay, we'll talk about both of these questions as we go along. Okay, this is an EM picture of the epithelium. It's a very nice one. You can see it looks like a toothbrush. You know these toothbrushes that are the automatic ones? They have a rotating. So this is, you see, they have these little cilia here, all these ends of the toothbrush. And this is the olfactory receptor, and these are these huge supporting cells, and the basal cells are little round things that live over there. Okay, wow. It's unbelievable, huh? Very sad. La la. Let's see. Okay, let's just a few slides and then we'll take a break. I'll talk fast now. Okay, there are millions of sensory cells. They're beating rapidly, asynchronously, so it looks like a jelly. And this I talked about 2 months of age, each receptor. The molecules that we can smell are all volatile molecules. Usually they're very small. We cannot smell molecules which are over 300 molecular weight. And normally the odors that we can smell are lipophilic molecules. And the first experiment started by punching electrodes into the olfactory receptor neurons. You can take the tissue out, put it in a dish, and stick an electrode into the receptor cells. And the first thing you can see when you blow an odor onto a receptor cell is that it has a depolarizing step and action potentials. Like I told you, it's a unique receptor neurons because it sends its axon into the nervous system. And in the 80s, there was some search for the olfactory receptor itself. What is the olfactory receptor? And the initial experiments were done by using patch clamp, looking for that protein, for the receptor itself. And when you give an odor, you can see that there's an opening of a depolarizing current. This is a cation channel, as you can see, because the currents are depolarizing. And in fact, people could recapitulate to some extent the responses of an odorant receptor neuron by inhibiting phosphodiesterase with a compound called IBMX, and this is well known to inhibit phosphodiesterase and increase cyclic AMP. And the search was, the trend was to find these G protein coupled receptors that operate through the G protein complex, as you may know already. And then the idea was that there is a G protein coupled receptor, which might be the odorant receptor. And people looked after that. And the story begins, it was connected, it was found that it was connected actually a little bit later on to this cation channel. We now know which channel exactly it is. is a cation cyclic nucleotide gated channel. When you knock out this channel from an animal, you get no responses. The animal is anosmic, as one can see here, 13 different odorants, flat in the knockout, and you get nice responses in the olfactory nerve when you record from these cells. And we're going to go to the break with the huge news in 1991, which shaked the field very strongly. So the lady is Linda Buck, postdoctoral fellow in the laboratory of Richard Axel. She just finished one postdoc in immunology and decided to go to the laboratory of Richard Axel to look for the odorant receptors. And she works for 8 years, no publications. 8 years she's working hard to find the odorant receptor. Okay, what is the receptor for odorants? We know that there are receptors for on the cones and rods for light, but we don't know the receptors for odorants. And what they find is an amazing discovery, which gave them the Nobel Prize, is that there's not one receptor. There are 1,000 receptors or 1,200 receptors in the mouse for odorants. Okay, it's a family, it's a multi-gene family of receptors, and this is a huge discovery because how many genes are in the genome? So the number is changing. Okay, when last year when I gave it, it was 25, but around, let's say, 25,000. Okay, the general ratio is not changing. So out of 25,000, 1,000 genes are for smell. 1,000 genes. This is an enormous capacity of the genome to dedicate for one system. How many receptors are for vision? 4, okay. 3 for color, 1 for black-white intensity. And here we have 1,000 receptors which are dedicated in the genome for smell. Actually, it's the second largest family. The other one is the immunoglobulins of the immune system. One can realize why we need an immune system, so many genes to fight against attackers. Okay, we need a lot of genome to go to the war against bacteria and other invasions of the nervous of the body. But 1,000 of these are for smell. And we're going to go to take a break, and we will go back to these 1,000 receptors when we come back. So 10 minutes. 1:05.
איך הסלייד הקודם היה? מה אמרת בו פסטיקו?
סוג נוקאוט? התעלה הזאת. התעלה הזאת.
כן.
Okay, the cyclic nucleotide cation channel. If you assume knockout, when you do a knockout to this channel, the whole sense of smell is
But what is this channel? If you're saying you have 1,200 receptors.
No. These are the receptors that detect the odor. And this activates G protein gamma cascade and opens the channel. That was well fast. Okay, but the big discovery is this.
It's a perfect G protein.
Yes.
So means 1,200 is also in mice, or is it the same?
No, it's not the same. We'll talk about that. Humans have 400 receptors functionally.
Okay.
We have 960 in the genome, but half of them are pseudogenes.
Okay.
Mice have 1,500, 20% pseudogenes. I will get to it here.
So means they're never expressed, or are they?
They're just unfunctional things. So if you look now, we know more and more genomes. This is like an update of 5 years ago. So the opossum has 1,500 olfactory receptors. Dogs have 1,100. Humans have 96, but 50% are not functional. multiple 700 and the
The
rest.
number is how many are non-functioning or how many are functioning?
How many are and how many are pseudogenes?
And how many are
So 400.
So pseudogene means that it's in the genome, but it doesn't produce
There is a
protein.
mutation which is needed for the stop code, so it's not
So the, okay.
it's not functional.
About this one.
Yeah.
So paradox, maybe it's a boring topic, but I still think it's a bit strange to compare the whole order space of all organs that exist in the world with the pure tones only, because pure tones is just a small subset of possible acoustic techniques.
But you can construct any stimulus from pure tone.
You can construct any strong molecule also.
Yeah, that's fine, but we have all kinds of constraints. We don't know what are the molecules, that's one thing. How to extract them.
Very hard to measure, and this
Yeah.
just conceptually, I think it's strange to say that the Odoron system is more complex than the tone system. Maybe technically, yes, it's more difficult to measure, but I don't see why it's a more complex concept. So on the one hand side, you have mixtures of chemical components, and on the other hand side, you have mixtures of pure tones.
Yeah, that's fine. But
Pure tones
again,
are
the molecules, how many molecules are there in the world?
Not a great number. You can make common change as long as you want, right?
No, but in practice.
In practice, nobody knows. It's true.
No, again, but it's a matter of putting it into a system that one can quantify, measure, and understand.
I agree that technically in the lab it's much more difficult than working with sound. This is not the question.
No, not that. I want to understand what is the range of capabilities of an animal to hear. So I can put it on this axis when I work with pure tones. I can say an animal cannot, a human being cannot hear above 20 kilohertz. That's a very clear cutoff.
True, but you can
In
also
other
use...
ends, it's very difficult. I'm not saying that it's not possible, but we are not there yet.
So
What Noam said is exactly the same thing. There's one axis of pleasantness based on the compactness of the others. We cannot detect, just like the axis of sound, we cannot detect molecules with a compactness over such and such.
So, but I would say that to me it seems most analogous to say, okay, in audition there's one receptor type, hairset, and in order sense there are, I don't know, in humans 400 functional receptors, so I would say that each of the 400 receptors is like hairsets, right? So you have 400 dimensions, and for each of the 400 receptors you can do the one-dimensional thing, right? You can increase the concentration until it becomes detectable.
No,
Decrease it,
it's a little bit more complicated. The hair cells is more uniform. Hair cells, they behave in a certain way, and we can really understand how they can convert sound based on frequency mostly and the intensity of movement of the vesicular membrane, etc., and how it's converted to spikes and perception, etc. That's the whole. That's what we're here for.
There are interactions in hair cells. It's not that hair cell here and hair cell here is completely independent, right? They are mechanically
Okay, but
coupled.
we can understand that. Here we will see it's a completely different system. They're all scattered on the epithelium. You will see that each receptor can respond to different molecules.
Yes.
Depending on the olfactory system, if you look at the general nervous system, it's highly dedicated to a given organ. So is it really a 400-dimensional space?
Depends on how much they interact, right? So maybe it's even much smaller.
It's probably much smaller, but now we're trying to understand these 400 different dimensions.
So
What
then
is
it's not
the
that,
rule?
it's not that the
Other
system
end-
is more complex, it's just that the research is more at the beginning. So audition
Yeah,
has been researched much more in the past than olfaction.
It's more complex to comprehend, or at least for us to understand.
Okay. It's not this whole question is not that interesting anyway, right? Because there are so many open research questions that you can deal with, and why should you care so much about the complexity if you have some work to do?
Okay. No, it's also about what you can or cannot do. Some people say, other world is so complex we cannot reduce it to a manageable thing.
Yeah, so concentrate on I don't know, non-cyclic, completely saturated carbon chains and research only them, right? It's like doing only pure tone stimuli in the nervous system.
Some people do that, yeah, but
Or only gray pictures in vision.
yeah, but again, if you look at pure tones, they are basic components of any sound that you play. If you look at free carbon chain molecules, they're not a component of any order that you have.
It's true,
Yes.
but it's a start.
No, no, I'm not saying people do that. We see exactly papers do exactly that. But the whole comprehension of the stimulus is not in our hands as solid as visual stimuli.
So in this, humans can discriminate more than 1 trillion odors. In this comparison in the paper, they compared the discriminability of us of pure tones to complex odors, right? So they put this number for how many pure tones can we discriminate, and they compared.
No, it's not a fair. They also said that it's not a fair. They said, look, people did a serious analysis of pure tones, and they found 400,000 pure tones. That's the number for perception. In humans, nobody did this work at all. Okay, and the number is 10,000. Why? Because they took the
like from the 20s.
exactly. They took 9 descriptors and whatever, and they came up with a number.
And
They say we have to do it like the auditory people, but now let's play with mixtures, not with single components. We get to trillion, and now the olfactory and auditory and visual people, you do it right yourself. And let's see who wins. Don't tell us that odors is less than, because this is the notion. The notion
It
is
used to
that,
be the notion.
that the smell in humans is a sense which is not as important, or not the perceptual capacity is much less. Basically, here they give them a teeth.
Yeah.
Because it's less difficult to survive. You can survive without it.
Yeah.
In evolution.
In human beings.
Yeah.
Yeah.
Evolutionary effects.
Yeah.
There are surgeries where you report from the brain, for example, epilepsy and other, and there are some tasks that they show to people, pictures of sounds, then they record from different areas. There's the same research have been done for odor, for
So for actually Parkinson's patients, they know that one of the first symptoms is that they lose the smell. They become nosebleed. A guy was talking to me a few times about this. Actually, there is a huge population of dopaminergic cells in the olfactory bone, which is extremely sensitive. And I think these are the first ones who die. I don't know why, and what's the connection. The guy said that there is there's a whole full theory that because the connection of the neurons that go into the skin, that the Parkinson the degeneration starts from the olfactory process.
It was known for people with Parkinson's.
Yeah,
How
but
much-
still,
how
if your
big is the
nose is
genetic
clogged
component,
in the winter, you can still survive well. And
But
pass
what
the
you can
day.
learn if you record from basal ganglia, at the same time you give different odors as a stimulus to a patient during the surgery. Can you learn something? What is the research that you can do? Because I can give for Parkinson patient, I can give just to
You're
call
asking
him
me,
come to me.
is it interesting to
Yes.
ask a patient if he can smell or not smell?
Yes.
So in many cases, I just don't think we know enough on that system. Okay, one thing that we found with Leo, our neuron paper from 2 years ago, is that the smells have a lot of neuromodulatory impact on other senses as well. So in the case of the mother and the pups, when she smells her pups, it impacts how she encodes the auditory information. Okay, when she smells them, she has a lot of more responses to the pup call.
So
There's a lot more that we will know because the information goes through the nose, and it spreads to cortical regions and has a lot of impact on all kinds of hypothalamic nuclei that have impact again. So the loop is closed very fast. The question is whether the basic depends how you ask the question. Because very basic perceptual decisions, I think it's easy to make. And you need a lot to screw them up.
I can tell you one thing. So there are some studies with humans and smell and fMRI, and there's this theory that when you have when you encode a certain memory, it's somehow called episodic memory, and smell something at the same time, and then later the smell is presented, that the smell alone re-instantiates a certain hippocampal activity pattern. And this has been done with fMRI. It's a paper by Jan Born, I think Science. And to do this with single unit recordings would be a big experiment.
But they need to record from hippocampus or from
Which is-
other
Which is
area which is
huge.
responsible.
No,
It's
but
not,
the,
it's
the
not a
factory
perfection
system.
experiment, but it's close.
Yeah. So it goes. We will also get to the circuitry later on, but it goes to entorhinal cortex, for example, directly
Then
from
to
the
the
body,
hippocampus.
and then to the hippocampus, etc. So it, it runs far and deep, but not so much to, to high cognitive tasks. So it doesn't go to the cortex like other senses which are normally tested.
Basic here we don't know as well,
I
right?
don't know.
But
No.
so from
Okay, let's start. I'll make myself a cup of water.
Some experiments, but
LAZ, only one.
To work with low dynamic information, you must ignore the risk that you cannot read through the paper.
What's the answer? Why are odors more conjugated? Lo que se está driving and
Okay, go for it.
אגב, בהקשר שאמרת שהריח הוא כן או לא ראשוני, בבני אדם בעצם הרבה פעמים זה הדבר היחיד שיש לך כדי לקבוע מהו משהו. אם אני אתן לך בקבוק עם שמן, תקשיב לו, תסתכל עליו, זה יכול להיות מסתבכים, מה צריך להריח? זה הכלי היחיד שלך לבדוק מים דברים.
לפני חודש שתיתי וויסקי. I was drinking whiskey and my son came out of his room. Daddy, this is apple juice. Can. Whoa, he's 10 years old. He was not very happy. I told him, you have to smell what you take from your father, especially if it's high quality whiskey. Okay.
It went down relatively easy.
Okay, big, huge discovery. 8 years of work, Linda Buck finds the family of odorant receptors, a family of 1,000 genes, making a huge impact in the field. First of all, it's the largest the largest gene family known. Most of the variation, it's a 7 transmembrane domain protein. From this family, most of the variation, you can see the highly conserved among the 1,000 proteins, is in the membrane, in the 7 transmembrane domain, and most of the variation is in the region where we think is the odorant detected, in the pore where the odorant is detected. We don't know much about that.
Where is the inside?
The inside is here, the outside is these loops out here, and probably interacting with these regions, but basically there's a lot of variation in these genes. Moreover, if you look where are the genes on the chromosomes, there's not some special clustering that one can say that all the 1,000 genes are in a particular place. In chromosome number 11, there are many chromosomes, there are many odorant receptors, but you can find them all over, okay? In chromosome number 13, only a few, but basically they're scattered all over the genome, these olfactory receptor genes. There's no apparent logic to their expression. Like I said, it's a 7 transmembrane domain from the G protein coupled receptor family, as we saw before. One thing which is interesting about these, which you should know, and this relates to Neil's question before, is that we call these receptors orphan. They're orphan because we do not know the ligands. This also has to do with what Johannes was talking about, because we don't know how many molecules are out there in the world, and we cannot find which molecule excites which G-protein coupled receptors. We do know them. If you look at other GPCRs, if you look in the mouse, we know almost 60% of the ligands of the known GPCRs, okay, starting with the receptors on the heart and going through many other receptors. I'm sure this number is higher and now, but not, we don't know all of the ligands, but most of them. For the olfactory receptors, if you look at, now this number in human is much better because of the paper I'll show you, but in the mouse there are 1,000 receptors almost, and we know the ligands for very few, okay? Very, very few. And why is that? How do you do this matching? How do you find the ligand, the molecule that binds to the protein? One can do a screening, okay? The companies of drug discovery, they're very good at this. There are very good ways in the industry, because there's a lot of money in medicine, to do screening. Let's say you find some protein which is important, and now you can, blow molecules at it to find which molecule activates this protein. There are very sophisticated ways to do this in the industry, basically to find drugs and their impact on this or that protein. But this was not possible in olfaction because we could not express olfactory receptors in heterologous systems where one does these screenings, okay? And this was changed in 2004, where they found a family of shuttling proteins, okay, that basically they shuttle you can express these olfactory receptors in hex cells, in heterologous systems, and they will be shuttled to the membrane, and now finally we can do our assays. And since 2004, it is now possible to express odorant receptors in systems like hex cells and try to look for their ligands. And indeed, at least for the human receptors, this has been done quite successfully. And we know more and more. I think we know in humans they have something like 400 olfactory receptors, and we know the ligands of about 40 or so, or maybe 50. I think at the moment, but still, this is to a large extent an orphan family of receptors. We don't know what is the ligand. Three things about this family of receptors. Yeah?
Is it always just one ligand per receptor?
No, we'll get to that too. You're faster than I am. There's a dramatic change in gene repertoire between different species, large number of pseudogenes, and a large variation within species. Again, this will get to your previous question. Okay? Let's look at the genomes. Now we know much more. Again, this is outdated. Opossums have 1,500 olfactory receptor genes. Dogs have 1,100. Humans have about 960. Rats now we know have not 1,000, but 1,500. Platypus, which is an important part in split of evolution, has 700 olfactory receptors, etc. In parentheses, you can see the number of pseudogenes, meaning that there's a mutation somewhere in evolution in the genes, and this gene is not functional. So basically, humans have about 400 or so olfactory receptors. Mice have 1,200, 1,200 olfactory receptors, okay, which are functional. So there's a large variation between the genes. Not all the genes are the same. We don't have the same olfactory receptors as dogs do, etc. Okay, and if you look at this table, and until now we talked only about the olfactory receptor genes. Again, humans 960, 50% of them are pseudogenes. And at the time where this was written in 2008, not all the genomes, or now we know many more genomes, and I'm not up to date. Just to tell you that there's a large variation and also a large number of pseudogenes. Some species, 80% of the genes are pseudogenes and not functional, and some only 15. An interesting phenomena, and this has to do with Neil's question, was found by Hiroaki Matsunami when he was looking at an odor which is called androstenone. Androstenone is an odor which I will give you, and half of you will tell me that it's sickening, and half that will tell you that it's vanilla. It smells like vanilla, which is a good odorant. And what they did here is that they looked for variation in they found the receptor that androstenone is activating. So this is one of the receptors which is not orphan. We know that androstenone activates this odorant receptor, and they looked for mutations in the genes in different people. And in fact, they can find a good correlation between the mutation. Actually, it's a 2-point mutations, one in the R and one in the T here in site 133, where people with this mutation will tell you that, let's say I have an RTRT, will tell you that the odor is sickening, and if they don't have this, they have a different combination of this mutation. In 2 sites in the odor receptor, the whole perception is changed. Okay, so basically, just one sec, this 2-site mutation in the odorant receptor can change the whole perception from sickening to vanilla. It doesn't explain the whole thing, but at least in part. Just one sec. This is a paper from 2 weeks ago, Mainland et al. Now they published recently the 1000 genome project. So one can access the genome of 1000 different people, and they can compare how noisy is the genome. They looked here, I think, in 27 different odorant receptors out of the 400 in human beings, and they can see that they are 70% alike, but there's 30% variation within a species. So your set of odorant receptors in your nose can be up to 30% different than my nose. Okay, so we have a personalized nose, so to speak, with different mutations. The question is whether these different SNPs, single nucleotide polymorphisms in the genes, can have a dramatic effect on the function, on how we smell things. This is not known. And actually, they did test it in this paper and found only that they can explain 10% of the variance. So this has to do with what you were asking before. When I go to a gas station and fill tank, I love the smell of the gasoline. My son can puke when he goes into me. You know, he goes like that into the, also to the spice store. If you go to the market, I love the spice store. My son can't stand it. Whether this is, you know, different SNPs, different variations of the receptor, etc., maybe like in the case of androstenone, but generally in the few receptors that they tried to explain, they could not really find a full explanation. They could explain, they say, between 10 to 15% of the variance to the strong odors that excite these receptors. Shoot.
Sometimes don't smells change over time. So your son is young, but maybe when
Yeah.
he's older, he'll like it. So they
Yes.
account for those changes.
Absolutely. So there's a lot, you know, the big trend now is epigenetics and things like that. We are changing all the time. We're on a constant slope. But again, generally speaking, on this axis of pleasantness, you know, a rose will, or an orange will smell like a nectarine in the same family, and it will be on the good side of the spectrum. And dog shit and cow shit will smell on the bad side of the spectrum and generally similar at any age.
In the case where you show the 2-point mutation, so the ligand would bind to the receptor in both cases?
Yeah, and we'll activate the...
Another kind of binding? I mean, what would...
Yeah, nobody really knows because they test this in hex cells, okay? And when you take a hex cell, it's a human embryonic kidney cell, which you can plate easily, and they connect it to all kinds of fluorophores, which show the activation of the receptor. But they don't know anything beyond that, okay? Basically, to really understand that, you have to express it in a neuron, record from the neuron, who knows, maybe also the next station, the synapse, how the synapse in the next station responds, etc. Here we don't know. There's a huge leap between the genetics and the analysis of the receptors in the hex cells to human perception. Okay. More very basic discoveries about the odorant receptor neurons and these ORs. First of all, as a rule, which is another amazing phenomena in neuroscience, each odorant receptor neuron will express only a single odorant out of the 1,000 receptors. Okay? So there are 1,000 receptors. Each olfactory receptor neuron in the nose will express only one out of the 1,000. This is a huge deal in gene control. People who study genetic control, how does the neuron knows to express only one gene out of the 1,000? Okay, it's a huge deal. In the last several years, there have been a few Cell papers, Science paper, and Neuron paper on how they do it. We will not go into it because this is for the course of genetics. But the bottom line is that one neuron, one gene out of the 1,000 of the repertoire. And the results that support this claim is that if you make a transgenic mouse and you express GFP in this transgenic mouse, which is coupled only to one receptor, let's say receptor 71 out of 1,000, so 71 GFP, and you look at the epithelium, you see 0.1% of the cells are labeled. That's the first indication that only 1,000th of the cells will express this receptor. Whether this is completely even among all receptors, we don't know because we didn't make so many mice. But the other mice that we have made, not we, but others, also show roughly 0.1% of the whole olfactory sensory neuron population will express this given OR. One. The second evidence is when you suck out single neurons and do single cell qT-PCR, or RT-PCR, sorry, you guys already know what PCR is, one finds in all the cases that only a single mRNA species of the odorant receptor is expressed. These two evidence suggest that each olfactory receptor neuron expresses a single OR. Shoot.
What amount? They all express the same amount or some variation?
I don't know exactly, but I don't think anybody ever tested it exactly. But generally, it will be good enough to be expressed on the cilia, and actually much more, because we'll see also something interesting about these olfactory receptors. Or maybe we won't. Yes, in enough amounts, yes. Actually, they express it too much. They express it also in the axons for some strange reason, which I will talk a little bit about. Probably not. Because we need to get to the computations and neuroscience. Okay. Yeah. No, don't be.
You said it's a genetic story, but what is, can you say a word about what is the mechanism that explains it, the difference in the transcription? What?
Basically, once the first, so first of all, it's a stochastic thing, which one will be expressed. But once this gene is expressed, it depresses all other genes in that family to be able to express.
The transcription, because you said there's not even mRNA.
No mRNA, yes, even the transcription, exactly. Okay, it shuts down all the other 999 genes. The big issue, Richard Axel, the one who found the odorant receptors, maybe I won't say this on. So basically, Richard Axel, who found this, had a huge Cell paper. A guy named Stavros Lombardos, extremely smart guy, big Cell paper. And 2 years later, they found that the mechanism that they found is completely wrong. And since then, Stavros got a position in UCSF, and basically he's working on that problem. He admits that his first paper was wrong, but now they found the mechanism. It's much more complex than people thought, but it's a huge thing in genetics. Anyways, there's a wrong paper in Cell, which nobody retracted, but everybody knows about it. Okay. Bon, if you look at the whole epithelium, in a strange way, it is divided into 4 different zones of families of olfactory receptors, about 250. in each zone. We don't know what this means, but if you look where is this a given olfactory receptor expressed, he will be expressed, let's say, in zone 2. Okay, only these blue cells, but there are about 250 in each zone. What it means, we don't know, but that's one fact. From the olfactory epithelium, the next station, like I told you, the axons are going into the next station of the olfactory system, which is called the olfactory bulb. In mice, it's a huge bulge in the front of the brain, shown here in red. In humans, it's down here. As you can see, the neocortex is something that in human evolution hugely expanded. If you look from the bottom, these are the two olfactory bulbs in human beings. In mouse, you can see that it takes a lot of hardware. Not that hardware always matters, but nevertheless. Okay? If you look at a mouse, a rodent, olfactory system, it is basically composed of 4 different organs. The one which I talked about until now is this olfactory epithelium. The mouse smells. You can see this is the snout of the animal. This is the nostril. This is the palate this is the tooth and the head palate, or the palate of the mouth, the upper palate of the mouth. So when the mouse sniffs, air molecules will go into these olfactory epithelium and will activate one of 1,000 different receptors. There's actually another group of tar receptors, which is called trace amine receptors. I gave somebody the TARS paper? You? Okay, so you're going to talk about the TARS, okay, which are expressed here in the olfactory epithelium, and they send their axons to the olfactory bulb, shown here in blue. In fact, there are 3 other organs. One is called the vimo-nasal organ, which we know something about. Yoram Ben Shaul, which will give you the 3rd lecture, will tell you about this. This is thought to be the region where pheromones and non-volatile odors are extracted. Humans don't use this, but rodents use this a lot. This is part of an active sniffing system. If you take an animal and you see that they're sniffing the head, and or dogs that they sniff themselves in the anogenital glands, they use normally the VNO system. It has to be an active pump. Yoram will talk about that. I won't going to talk about it at all. Just they send their axons to these accessory olfactory bulbs, shown in green. There are 2 other ones which are cool. One is the Grunberg ganglion. These are all Nature papers. Everyone that found these is a big thing. They are expressing 5 TARs and a few vimo-nasal receptors, and they are thought to be alarm pheromones. It's a small structure right on the entrance to the nostril. There's another one here which is called the septal organ of Mazera. Anyway, and this has 120 out of the 1,200 ORs, and they are sending their axons to these small targets here. We're not going to talk about these at all, only about the main epithelium. And Yoram will give you a lecture about the pheromones, because it is highly cool and sexy system, which triggers behavior very nicely. Okay, this is exactly the same thing like you saw before. That's in cartoon. This is the real thing. Okay, if you take an animal, this is a rat, and you cut in the middle of the head, you can see the entrance to the nostril. And look how huge. This thing is the shaky one. If you look at it, it will dance all the cilia here. This thing, they will be like a jelly. They are beating here on all these huge epithelium. And this is the olfactory bulb. The whole brain is something like this of the mouse. Okay, it ends here. This is the brainstem, cerebellum, cortex, and that's it. Huge olfactory bulb and a huge olfactory epithelium. So it's a huge sensory organ in terms of its size. And one remarkable finding, which really taught us quite a bit on how odors are processed, is that all the olfactory receptors that express a single gene will converge all of its axons into a single spot in the olfactory bulb. Okay? So let's say we have, I don't know, something like 15,000 or 20,000 olfactory receptor neurons that express olfactory receptor 700. All of these will send their axons together. They're in this zone, but they will send their axons to converge onto a single spot in the next station, in the olfactory epithelium. This is shown schematically on the top here. You see, let's say, from zone 1, which is a little bit larger, all the neurons expressing the red receptor will converge onto the olfactory bulb, and all the ones who express the purple receptor will converge into a different spot. These spots are called glomeruli. One sec. Okay, and they all send their axons to find a single spot. This is just kind of a immunoreactivity of these axons. This, before I give you to the floor, this poses also, like the one gene out of the 1,000, which is a huge genetic enigma, here too. How can all these 1,000 axons migrate, find their way exactly into a specific spot? So in developmental neuroscience, the axon pathfinding of the olfactory system has been like a gold standard to understand. Okay, because normally we thought in the retina, I don't know if you learned anything about development already? Nothing about development of the nervous system? On the visual system, gradients, Sperry, chemoaffinity theory, nothing. Okay? Anyway, this is a different kind of story. Normally, if you look at how the neurons in the retina find their way up, there are beautiful gradients. Neurons from this side will go to the other side, and they use chemical cues. Here it was a mystery because there are 1,000 different glomeruli, and each one has to find its own spot exactly in the region. How this is done exactly, it's a very interesting developmental neuroscience question. Now, the floor is open for questions. I'm in deep trouble. Shoot.
Two questions. First one, we did in the research methods class, I did the data analysis to meet
mitral cells.
Yeah.
Yeah, we didn't get there yet. Those are the ones who read the input from the ORS.
Okay, so
one synapse away.
Okay, and the first question is, so these are all single neurons that go to P2, or is it?
Yes, these are P2 neurons. This is the name of a receptor out of the 1,000. They express some kind of marker. In this case, it doesn't matter, which gives you a color. And all of these are 15,000 axons or 20,000 axons.
They're being also regenerated, so they need, like, it's
yeah.
constant. The one dies and the other
exactly.
Finds, like, generates and needs to find its location.
Exactly. Continuously, the axons have to find their way to the right spot, and it's amazing to see how accurate they are. There are no mistakes. One at a time. Ladies first.
It happens each time there's a regeneration of the neurons. You said that the neurons every month or something like
yeah.
die.
Yeah. So basically, if you look, you know, not all of them die at once, and some neurons will live forever through the whole life of the animal, but some there's quite a bit of regeneration. So they would say the average is 1 month. Some will die after a few days. Some won't make it and die. But basically, they have to maintain this network continuously. Okay.
I can see that the zones are constrained from the epithelium to the bulb.
Yes.
And so is that what we see here? Is that the way it is?
Yes.
It's known as accepted?
Yes. We don't know what it means, but these zones are conserved. Actually, this is a developmental thing. We know that they are put, when the nervous system is put, it's put zone by zone. First zone 1, and then zone 4, up to zone 4, from 1, 2, 3, 4, but we don't know if there's a functional meaning to the... Yeah.
Do these neurons have any synapses on the way?
No, this is just an axon going all the way through and making a synapse on its target. No, there's a branch of axons with different synapses on it, but only here. Only here. Here, no synapses at all. It's all myelinated axons. They go together and they synapse into the spot, which is called the glomerulus. Yeah?
These 4 layers is just in rodents or also in humans we can see?
I don't think we know it in humans at all. What we know is 99% in rodents. So the zones are preserved. We don't know what they are. But again, all the axons, they go and they terminate here. Okay. Okay, just a few more points, fast points, to make things a little bit more complicated. There are actually 4 different glomeruli of one kind. So if you look at an animal, you will find 2 glomeruli in each olfactory bulb which express a given receptor. Let's say receptor number 805 will have 2 copies in one bulb and 2 copies in the other bulb. And people claim there is some kind of symmetric functional symmetricity here, so that the distance between the 2 different receptors is equal. Again, we don't know why we have doublets of this in each olfactory bulb. The projections are ipsilateral. Okay, and moreover, there are neurons that are connecting the same receptor in the different bulbs. So why we have 2 ears, we know. Okay, we can locate things in space. We will learn about this ITD, ILD, whatever, in the next session with me. Why we have 2 eyes also, you know, we can cover more space, we can look in 3D, etc. Why we have 2 nostrils, nobody knows, but the matter of fact is that these 2 functional units, because this is a functional unit, okay, the receptor that expresses in a given spot here, in each side of the brain, is a functional unit, because it will respond to the same odorant, to the same part of the world. They are also connected in extremely precise way via a second-hand circuitry. So there are neurons here. These are not the mitral cells. These are the mitral cells. There are neurons here which connect these 2 functional inputs between the left and the right hemispheres. What is their role? People have all kinds of ideas about synchronization and how it's important, but nobody really knows. Okay? There's a paper I will touch upon about rats smelling in stereo. Again, just the title made it a science paper, but okay, so to sum up, in olfaction, the stimulus is a chemical one. Vision is light. I put this because this is like the well-known column, and this is not. It works through an ion channels, which I went out when I went fast briefly. Okay, the C and G channel. It's a cyclic nucleotide channel. This is goes through cyclic AMP. In vision, it's cyclic GMP. I don't know if Shaul talked about the molecules and the rods and cones a little bit. Okay. Anyway, there it was GMP. Here it's AMP. He surely talked about hyperpolarization, right? Which is very different. Here it's depolarization. The receptor neuron. This is unique. It projects to the brain in olfaction. We use action potentials here. There's graded potential, and at least in mouse, huge difference in the number of receptors and genes. Okay. Now let's go to the mitral cells. One thing is that if you look at glomeruli, these structures are noopile regions where synapses are made between the olfactory receptors from the nose and the projection neurons from the olfactory bulb, known as mitral cells. Okay? So mitral cells will send a single dendrite into here. All olfactory receptors of a given type will send their axons into a glomerulus, but there will not be intermixing. So you cannot find a glomerulus which receives input from 2 different receptors. Okay? So all receptors send axons to one glomerulus, but each glomerulus receives input from a single olfactory receptor type of neurons, about 15,000 cells. Now, Linda Buck finds these receptors, which gives her the Nobel Prize in 1991. It's her paper from 1991. Then she goes on to Harvard to have her own position. And in 1999, she starts to look at the coding possibilities of these receptors. How do they code? This is the first paper, a cell paper. What she's doing is she takes 2, 4, 6, 8 different odors, different types, alcohols, acids, in this case, and she isolates 14 different olfactory receptors, 14 cells, and does calcium imaging in a dish. And what she finds is that when she, and this is the human descriptors of these odorants. So this is proof that these are really different odorants, not only by their chemical structure, but also by human descriptors. And what she finds is what she calls a combinatorial code in olfaction. What does it mean? If you look at this odor, he will excite in this panel only odor number 5. Only cell number 5, sorry. But cell number 5 is actually excited by 1, 2, 3, 4, 5, 6, 7 different odorants. Okay? And if you look at cell number 14, will be excited only by this out of the panel of the 7 odors. So this is a very small panel of odors, very small panel of randomly selected olfactory receptors from the nose, but you can already see from this description, which holds to this date, is that the code is an combinatorial code. And from this, we learn 2 canonical lessons in olfaction. One is that different odorants, you can see from here, different odorants can excite, because statistically, if you choose 14 out of 1,000, these are completely different, will express completely different receptors. At that time in 99, she doesn't know which receptor is expressed on each neuron, okay? But it's probably random. But from this, you can see that different odorants will excite, or a given odorant, sorry, will excite different receptors. Some will have a wide impact, some will have a small impact, like this one. Maybe, but maybe this is just a random selection. That's one thing. Second thing, a given odorant receptor, because we know that each neuron expresses a single receptor, will be excited by a variety of odorants. So it's not one-to-one, but there's actually a combinatorial code. So if you look at a given odorant, 2 different receptors, they can be excited by a single odorant is typically recognized by multiple receptors. So let's say this is odorant, whatever, X. You can find different receptors that will be activated by it. Single receptors can recognize multiple odorants. For example, receptor X can recognize this molecule, but also another molecule. Okay, let's say the first 2 top molecules it can detect, and some will not detect it by any, or not by these 2. This brings us to a canonical concept in olfaction, which is different odorants are recognized by different combinations of receptors, indicated that the olfactory system uses a combinatorial coding scheme to encode the identities of odors. Okay.
Has anyone tried to find what is common to all the odorants that activate a single?
No, in terms of what motif on the ligand, we don't know all the ligands. Ligands is an endless world, okay? Unlike auditory, which we can break it nicely into its pure tone components. This is a very difficult task, okay? Very expensive task also. You want to get pure odorants, you have to buy them in sigma, and of course, you're always in a very limited part of the world to the animal, one thing. Second thing is until, 10 years ago, we couldn't even express them. So one has to decide that he wants to do the experiment and express this odorant receptor in heterologous systems to start to understand. It's a huge mess in that sense. And also, why is odorant number 700 more interesting than 900?
No, but what I thought that if you find some kind of property of the ligand that makes it bind, then you find the important property of that.
Yeah, but one has to choose a receptor to do that. Which?
You just say that if you find it for one receptor, maybe it will be the same property that reports.
So I would say no. We will see that a little bit later on, because some receptors have very narrow receptor fields. Others are wide, so maybe one has to choose one with wide and one with narrow, but still we don't have access to all the stimuli. And certainly we don't know which one is which. It becomes a very complicated issue. And when you want to choose what you're going to do when you grow up, you want to choose something which is